Hey, didn't see you there. It's James.
Hi, I'm Emily.
Hi, I'm ready.
It's your boy, Brandon.
Gene silencing happens when a gene is turned off through adding RNAi or the RNAi interference system.
This RNAi system has control over which genes are active and how active they are.
In this experiment, we're causing a mutation in the C. elegans by silencing a genome.
We will achieve this by introducing bacteria containing double-stranded RNA into the C. elegans diet. By doing so, we will trigger the C. elegans RNAi, thus preventing gene expression.
You may be asking, what even are C. elegans? I've certainly never heard of them or participated in a concurrent enrollment biology class at RCHS. Well, C. elegans are near microscopic members of the Nemata phylum. To put it plainly, they're a really small, worm-like species. We need to address why we even use C. elegans in this experiment. Well, we use the C. elegans because they make ideal model organisms. Their DNA is very similar to humans, they have a short lifespan of 3 days, and they reproduce frequently. As an added bonus, they live off bacteria, making it easier to apply the double-stranded RNA to silent genes into their systems.
L1 wild types are just a little bit larger than the eggs and are very, very small. The L2 is similar to the L1, just slightly larger. The L3 is beginning to show traits similar to that of the adult and is larger than the L2. The L4 looks exactly like an adult, just slightly smaller, and it doesn't contain eggs. The adult is a fully grown C. elegans. It's the largest of all the different life stages and has eggs.
We cause variation through this process of chunking, where we transfer the N2, which is the wild type, onto a feeder plate containing the RNAi-inducing double-stranded RNA bacteria, causing the variant of the dumpy. We aren't turning a gene on, but instead turning a gene called DPY11 related to growth and development off. We utilized ampicillin, the purpose of it being to kill any bacteria that didn't contain the dsRNA. This was to reduce the risk of certain worms not having the dumpy variation due to eating the wrong bacteria. Also, another huge factor used to ensure dumpy variant was IPTG. IPTG is a transcription factor that attaches it onto the promoter regions of the elegans, then transcribes the RNA, making it dsRNA. This makes it more likely for the C. elegans RNAi defense system to occur, because there's more bacteria with the dsRNA. Right here, you can see the dumpy mutation is a close, non-fatal variant to the wild type. They're shorter, fatter, and slower, but the defects won't cause any injury or fatality. You can see the movement type is a little different because they're inhibited by their shortness and fatness, which causes them to move slower and not efficiently.
The dumpy mutation is first observable around L3 to L4. Prior to these stages, the C. elegans are too small for any real differentiation to be noticeable. As you can see, we clearly have the L1, the L2, the L3, the L4, and the adult clearly shown. We were assigned to silence the DPY11 gene. Its function was to create thyrodoxin-like proteins. These proteins serve for morphological maturing as well as body and sensory organs. The results we observed, that being small, dwarfed, and sluggish C. elegans, is fully supported by the silencing of this protein creation. When we provided bacteria containing this double-stranded RNA, the C. elegans would eat and digest it. The double-stranded RNA would enter the cells of the C. elegans. The RNA interface would be triggered, believing it to be a threat. The dicer, an RNA tool, so to speak, chops up all the double-stranded RNA into shorter siRNA. Those siRNA bind to several proteins, forming an RNA-inducing silencing complex. The siRNA form a single-stranded and probe the cell for mRNA, which would be in our case both the natural C. elegans DPY11 and our fake dsRNA. The RNA-inducing silencing complex would bind and cleave it, making it no longer usable. As a result, translation would never occur and the protein thyrodoxin never formed. The protein for growth wouldn't be present and the C. elegans would never mature and stay as dumpies.